It's time to talk about cytology now. Moving one step up, we're going up from molecules to what molecules make, which is organelles and cells. So, next step is to talk about emergence with cells. So, emergence in cells relies on subcellular components. Those would be organelles. Subcellular components provide critical cell processes for the cell. So let's summarize what we're about to talk about here. There we go. Cells are the ultimate emergent system. They're very complicated, much more than cars, and they are the biological basis for the bottom level of life. Nothing smaller than a cell is alive. So they've got something special going on. Each organelle provides some critical function for life, and if you take the organelle away, the cell can't survive. So, bullet point: raw energy capture and processing, that's the chloroplast. Chloroplasts do things similar for cells that a coal mine does in obtaining energy for a community that is used to provide electricity. The chloroplast is a raw energy processor. Energy processing into something useful, that's mitochondria. Mitochondria are similar to an electric power plant. They take the coal, that's basically the sugar manufactured by the chloroplast, like a city takes electricity, but the electricity is being produced by processing coal, which came from coal mining. The water reservoir, that's a vacuole. Vacuoles are just big water bags to provide the cell water in times of dehydration. Important production facilities are ribosomes. Ribosomes are similar to a factory in that they produce machinery, like literally robots. Enzymes are somewhat like robots in that they're complicated little gadgets that do all kinds of little jobs for the cell. Cells also have transport systems, that's rough ER, which is similar to roads in a city or a town. They also have destruction capability, which is smooth ER, which is similar to like filtration equipment in an air conditioning system, in that it takes bad things out of the system. They can modify already produced materials. That's what the Golgi apparatus does. The Golgi apparatus is similar to a robot assembly plant, whereas the ribosome is in robot part production. And lastly, the requirements of the cell are a lot like those of a city. You got to have energy, got to have materials, got to produce them, and you got to process them, got to have water, etc., etc. So let's talk about some of the pieces in detail. Ribosomes, they're really small. All living cells have to have them. They're only about 23 nanometers, so they're the smallest of all the organelles. They're 23 nanometers in diameter. They're roughly 50% RNA and 50% protein. And they are the sites of protein synthesis. They are where the genetic code unfolds into an amino acid sequence. They create the primary structure of a protein. The secondary is usually on its own. The tertiary level, that's often Golgi or self-assembly, and then the quaternary is always Golgi. So here's what a ribosome in an artist rendition looks like. There's two pieces, a large and a small subunit, and they're shown. We talked about these in translation of protein synthesis. The small subunit attaches first, followed by tRNA, followed by the large subunit. It's the large subunit that has the peptidyl transferase that bonds amino acids together. So ribosomes are one of the basic essential components of cells. The next thing here is the endoplasmic reticulum. Endoplasmic reticulum, the ER, is a compartmentalizer. It basically divides the cell into spaces. There are two types, rough ER and smooth ER. This is smooth here, and this is rough here. Okay, the rough ER serves as a site for protein assembly for those proteins that have to go to the Golgi. So there's a lot of little ribosomes attached to the rough ER. They have a receptor on their surface, and they actually pick up proteins. They pick up messenger RNA that has specific leader sequences, which indicate in the scheme of things that the protein is destined for either export or special modification. So the proteins are made by the ribosomes. They're shoved into the ER. They travel through these sacs, and eventually they get out to the edge. There they bubble off, and then they go to the Golgi, and the Golgi then modifies them. So the ER compartmentalizes. It does provide a slight amount of mechanical support, protein synthesis location, and intracellular transport. In addition, smooth ER actually detoxifies substances. The Golgi, which interacts with the ER, is a membrane-bound structure that consists of flattened membrane sacs. That's this here. The Golgi's job is to package proteins that have been sent to it from the rough ER. The Golgi also transports vesicles, they're called, of proteins, which are small bubbles, to the cell membrane. The Golgi modifies proteins at the tertiary and the quaternary level, and the Golgi also produces bags of enzymes called lysosomes, which break down worn-out organelles. So those two organelles really closely work to each other. They share a lot of phospholipid membrane material because of that. Next one to talk about is the powerhouse of the cell. That's the mitochondria. Mitochondria are tiny organelles that are about maybe a micron in diameter. Here they are in a drawing. This is what they look like under an electron microscope. What you see here is inner membrane that's highly folded, and then a smooth outer membrane, and that's what this is showing over here. The mitochondrion's job is to produce ATP essentially by taking in pyruvate. Mitochondria they also have their own DNA, they have their own genomic DNA, and they produce a number of their own proteins. It's believed that at one time were separate life forms, and now they're living intracellularly inside of every multicellular organism on the planet, processing energy aerobically. Remember, pyruvate comes in, the Krebs cycle's in here, hydrogens are sent to the inner membrane, the electron transport system there processes the hydrogen into water, but in so doing, it manufactures ATP, much of which is transported, out to the cytoplasm. So mitochondria are famous for ATP production. They're also the source of carbon dioxide from the Krebs cycle in there. So that finishes the mitochondria. Let's talk now about lysosomes, or just bags made by the Golgi that contain enzymes that are important to destroying worn-out organelles. So they're bags of enzymes important destroying non-functional organelles, and they also digest specific macromolecules. They are also important to phagocytosis. If a cell is eaten, like this bacterium, it's absorbed into what is called a vacuole. This is a food vacuole, which is different than a water vacuole. When that happens, a lysosome fuses with it, and the enzymes of the lysosome destroy the bacterium, and then the bacterial chemicals diffuse out into the cytoplasm and feed this organism. So this is a single-celled organism that eats bacteria by phagocytosis. Bacteria is brought into a vacuole, lysosome fuses with it, hydrolysis occurs, and monomers diffuse out, and then the unusable materials are exocytosed out of the cell. That means like reverse phagocytosis. In animals, vacuoles are often food vacuoles. In plants, they're water vacuoles. In plants, the vacuoles like this store essential enzymes for some parts of the cell. They also store pigments for parts of the cell, and some toxins are stored in here that otherwise would harm things in the cytoplasm. Let's see, now that brings us to the next organelle, which is chloroplasts. Chloroplasts are like the coal miners. They are producing a raw energy source for the cell. They also have their own DNA. That's chloroplast DNA there. It's bacterial DNA in similarities. They have their own ribosomes. They manufacture many of their own proteins. They are believed to have been at one time separate organisms, and they were eaten, but they lived inside of their host cell. So the outer covering of chloroplast is double, unlike the mitochondria, which basically is two membranes. Chloroplasts have three sets of membranes. They have an outer membrane, they have an inner section of the outer membrane, and then they have the thylakoid membranes, which are on the inside. So the outer covering of a chloroplast consists of two membranes. Inside the chloroplast is another series of membranes in the flattened sacs of the thylakoids. We talked about those in a previous chapter. Chlorophyll is what makes these green. The thylakoid membranes have the proteins that produce the NADPH, that's reduced NADP and ATP. The fluid around the thylakoids is the stroma, that's this tan stuff going around the outside. That has the enzymes that produce carbohydrates. And with all of these pieces on the inside of the chloroplast, as long as light's coming in, the light and the dark reactions can take place, and the chloroplast can make sugar. And so this is showing water being used by the light reactions. Oxygen is made as a waste product. The dark reactions really don't make any waste, but they take carbon dioxide in, and using the hydrogen and ATP from the light reactions, they crank out carbohydrate, which is like coal. Carbohydrate is not directly usable by organelles, but it can be turned into ATP energy, which is directly usable by organelles. Which now takes us to locomotion. Cells have to have some way to move, and they do. Cells sometimes have lots of little hairs on their surface. The hairs are made of proteins. The proteins are called microtubules, and when they're really small and short like that, they're called cilia. So these are cilia, which are microtubules that are really short. And this is showing a paramecium swimming. As the little microtubules or the little cilia reach forward, they pull back and they propel the ciliate forward. There is another strategy that microbes employ, and that is the flagellar strategy. In this case, some cells have just one or two long microtubular projections. Then it's called a flagella, or flagellum is singular. And the flagellum is usually used to pull, but sometimes to push the cell. Sperm push with their flagella. Lots of unicellular prokaryotes pull with their flagella. So cilia, basically, the difference is cilia are short and numerous, flagella are long and few, and they both are made of microtubules, which are hollow pipes constructed of microtubes, which are able to then bend and propel the cell through the water. Microtubules are also part of the intracellular framework of the cell. They're all over the inside. They form a protein matrix to add scaffolding to the inside of a cell, and they actually reinforce the inside of the cell. Mitochondria actually, remember, supply the ATP to power flagella and cilia. Microtubules are different than microfilaments. Microfilaments are for shortening and lengthening cells, and there are small amoeboid organisms that can crawl using microfilaments, but microfilaments actually change the shape of the inside of a cell. Microfilaments are also responsible for human muscle cell contraction. The difference between these two is microtubules are usually outside of a cell, used to propel the cell through its medium, where microfilaments are inside of a cell, and they're for changing the shape of a cell. Muscle cells change shape when microfilaments shorten. And amoebas and white blood cells, when they crawl, they're changing shape as the microfilaments cause that. So in summary, what we have then is a story of how organelles, the plasma membrane, the cytoplasm, and the organelles contribute to the overall specialization and functioning of the cell. The cell membrane allows for exchange in and out of the cell. Mitochondria use the oxygen and glucose provided through the cell membrane for food combustion. So that's these little guys. It's not directly glucose, though. The cell has to crack it into pyruvate first, but carbon dioxide waste from the mitochondrion goes out through the cell membrane. That comes out. And then the nucleus produces all, this core right here makes all the RNA that comes out here so that the ribosomes can make the protein to build all this infrastructure in here. And the Golgi and the ER are sending and processing the proteins for all of the cell activities to take place. So now it's time to move on to DNA and how DNA is involved in cell specialization.